So again, like I said, I did turn from the human to the mouse for practical reasons. And I think a couple of the, voila, eureka moments that happened here. The first one, I think, was when we had discovered this system that I had started working on at DNAX, this receptor called NKG2D. It's a receptor that activates T cells and NK cells. And my postdoc who was working at DNAX and then joined me here at UCSF, named Heidi Cherwinka, she cloned the ligands for this receptor. And they're really interesting because in mice and humans, we have about 10 genes that encode these ligands for this receptor. But those genes are silent in healthy tissue. But they become turned on in cancer cells, virally infected cells, and they become upregulated in some autoimmune diseases. So that really set the stage for much of what I've done since being at UCSF. We had a receptor that's on all NK cells, it's on all gamma delta cells, and on human CD8 T cells. So when those cells, those immune cells, are circulating through the body, typically there's no ligand for that receptor in healthy tissue. But either cancer or viral infection or autoimmunity can cause the expression of those ligands on the surface. It's good if you have cancer or have a viral infection. It's bad if you have autoimmunity. So we've set up several models, autoimmune models, viral infection models, and cancer models to show the importance of that receptor system.
In layman terms, describe the ligand.
Okay, the ligand they're a protein that is not expressed on healthy cells. When cells become unhappy and they're disturbed by infection or by cancer, they then will put this protein on the surface of the cell like a flag, saying, something's wrong with me, I need to be gotten rid of. The receptor on the killer cells can now see that flag, know that that's the cell to take out. So there's a variety of ways that their kind of stress can do it. Many types of stress of the cell causes that process to turn those genes on.
Now, it would sound like a simple-minded person would say, well, you've cured cancer.
We've cured cancer in a lot of mice. That's the interesting part. Both viruses as well as cancer cells find a way to escape this mechanism. And one of the, another good surprise was when we started working with a postdoc here, Courtney Crane, who was working with a neurosurgeon here. We were looking at glioblastoma patients. Now, most cancers express these stress ligands. And if I put these cancer cells in a dish, the killer cells will kill them. But obviously, that does not happen in the people who have cancer. So when we started looking at the blood of these people who had cancer, we saw the receptor was not on the cell surface of the killer cells. It was gone. We're going, well, why is that happening? It turns out that when the ligand engages the receptor, it's downregulated, okay? And it makes it internalize and disappear. But we says, well, how do the blood cells, how do they get into the brain to have access to the glioblastoma to then downregulate the receptor? It turns out that the glioblastoma cells secrete a soluble molecule, which then make the person's own healthy monocytes make the ligand. So now you've got decoys. You've got normal healthy cells putting the ligands on, which are preventing the killer cells from attacking the tumor, because now a lot of healthy cells have the ligand and cause the downmodulation of the receptor. That was a big surprise, and we're now working together with Courtney and Andy to try and understand whether this can be a diagnostic use, or if you could stop the tumor from putting the ligand on the healthy cells, maybe the killer cells would find the glioblastoma and get rid of it. So we also had worked on several viruses which have ways of stopping cells from putting those ligands on the cell surface. So half of us are infected by a virus called cytomegalovirus. Once you're infected, you have it for life. It's like herpes, it causes cold sores. Or like chickenpox, once you get it, you have that virus forever. Well, CMV, if you infect cells with CMV, the infection causes the infected cell to start synthesizing the ligands for this killer receptor. What happens, though, is the virus, CMV, in both mice and humans, makes proteins that then grab and capture those ligands inside the cell so they never make it to the surface. So my first PhD student here at UCSF, Melissa Ledon, she's now an assistant professor down at UC Irvine, she identified some of the CMV genes that allow the virus to stop this process from working. It grabs the ligands and keeps them inside so the killer cell can't see that the cell's unhappy. So that's been keeping me busy at UCSF for a decade. Part of what's keeping me busy.
Okay, talk about the other parts.
Okay.
Other than grant proposals.
Yeah, I'm a professional writer. I guess, the other thing that I've been interested in is why do you have natural killer cells? Now, they were originally discovered because of their ability to kill cancer cells, but, cancer usually happens in old people because of accumulation of genetic mutations. I think natural killer cells really arose to deal with viral infection. There are a few humans which have no natural killer cells but are otherwise have normal immune systems. Those people suffer certain viral infections. That's what they end up in the hospital with and what has typically killed them. It's a really rare disease, but it's instructive that that's why these cells are there, is to deal with certain viruses. And herpes viruses are what NK cells like to recognize and attack. So we've spent a lot of time working in the last 10 years on cytomegalovirus in humans and mice. And what we've in the mouse, we're taking away receptors experimentally and manipulating the virus to see how it works. But we've also had the opportunity to work with clinicians. And one of my fellows now, Gundolin Meno, has been working on 2 people who have no NK cells to find out why they don't have NK cells. Those people have chronic problems with herpes virus infections, and one of the women has become infected with papillomavirus and has acquired cervical cancer because she can't take care of viral infection. So that's been a lot of fun. I think we've just figured out why these people have no NK cells. I think we found the mutant gene, because today, for less than $1,000, you can sequence all of the exons in your genome. So we got blood from family members and from these 2 patients, sequenced them, and I think we have a good clue now. And we're trying to validate that and prove we have the right gene. But that's been fun.
Anything else?
The other thing I think that we really found that was surprising was that natural killer cells had memory. Also that they could be completely virus specific in some cases. So before it was thought that NK cells, were non-specific. They would just kind of kill any cell that had, didn't look right. Largely through this NKG2D pathway being one of the major ways they would do that. However, one of my fellows, Hisashi Arasi, about 2002, he identified a receptor on NK cells in mice that allows them to specifically attack cells infected with cytomegalovirus, and he cloned the viral gene they recognized. So this is absolutely specific, which is a complete surprise that NK cells could be specific for CMV, have a receptor that would not see any other virus. The other thing we found that was surprising was that once these NK cells recognize and respond to CMV, CMV stays with you forever. If you're infected at 5 years old, if you live to be 100, you'll still be infected with CMV. So you've got to keep it in check for 100 years if you're lucky. And NK cells are part of that process. In mice, Hisashi found this receptor, which specifically sees CMV and controls it. So Joe Sun was a fellow who started working with me about 5 years ago and says, well, what happens to those NK cells after they see the CMV? We know they kill the infected cells, but the virus hangs around. They've got to keep it in check forever, for the life of the mouse, like they do for the human. So Joe did a really clever trick. He took NK cells that have this specific receptor called Li49H and put them into a mouse that was in all other aspects completely normal but lacked that receptor. And we knew they would die of CMV. He could then track the fate of those NK cells. Previously, it had been said NK cells only live for 2 weeks. Joe put the cells in, these mature NK cells with this CMV receptor, into the mouse, infected with CMV. Cells go crazy, control the virus. But Joe came in one day in my office and says, it's 3 months later, and I can still find those cells I put in 3 months ago. Said, they didn't die in 2 weeks like the textbook said they were supposed to do. So that was, our wow. NK cells are long-lived. We studied them and showed that they would then respond better the second time they saw a virus, saying, like B and T cells, they remembered. And they were also long-lived. Now it suggests that maybe you can vaccinate NK cells like you vaccinate other components of the immune system. So that was about 5 years ago, and that's what's been driving the lab since then. The most fun thing we're doing now, I think, is we've shown that that also occurs in humans. I think we found the receptor for human CMV and are trying to prove that that's true and come up with strategies to see if we could vaccinate your NK cells to make them work better against CMV.
How would you imagine you would do that?
Well, I think we know the receptor that humans use to recognize, human NK cells use to recognize CMV. We don't know the viral ligand yet. So that's the grant I'm writing this morning, is to try and identify that. If we could find that, like you do for your flu vaccine or your chickenpox vaccine, we could inject that ligand with a tasty adjuvant, expand the population of NK cells that would see the CMV infected cells and protect. That can be important in transplant patients. Also, 1% of human babies are born with CMV infection. And some of those go on to have hearing loss and they have developmental abnormalities. If you could vaccinate the moms, then you could prevent the kid, because they usually get it during birth from their mother. So I'm hoping that's the significance of my grant section, is hopefully we could stop this from happening to kids.